Hello, everyone. We need to start our unit on genetics, the study of how characteristics are passed from generation to generation, from parent to child, and then from that child to their children. And so the field of genetics was established by a gentleman named Gregor Mendel in the 1800s, and we'll talk more about his work in just 1 minute. So, genetics, it's the study of heredity. How are these characteristics passed from generation to generation, from parent to child, child to child, one after the other? Well, how it's done is actually through DNA. The characteristics are passed physically from parent to child. Don't forget that chromosomes are condensed DNA. It's a long strand of DNA that condenses into this little package called a chromosome, and that's what you actually give your child. And then this DNA is the genetic material that causes the brown hair or the brown eyes. These are the characteristics that go from parent to child, and the instructions for those characteristics are held inside of the DNA. So in class, we took a section of DNA called a gene, and we expressed that gene through transcription and translation. The A, T, G, and C, we transcribed that into messenger RNA. We then translated the messenger RNA into a protein. That process of protein synthesis, transcription, and translation. That's why it says these genes, or the sections of DNA called genes, code for the proteins that cause our phenotype. You say, well, what's your phenotype? Okay, your phenotype is the physical representation, what you see on the outside of the organism. It's the expression of the genes. So when you have a gene, and that gene is expressed through transcription and translation, and you produce a protein, that protein causes the phenotype. So I have brown eyes because my biological parents gave me a gene that my cells then read, transcription, translation, and produce a brown pigment. That brown pigment is then stored in my eye, and I have brown eyes. I then pass that same gene on to Emily. Emily's cells read those genes, or read that gene, produce that protein, and she then has the same eye color. So our phenotype is the result of protein synthesis, transcription, and translation. What you see physically is an expression of our genes. We are the product of the genetic instructions. Where did we get these genetic instructions? We got them from our biological parents. So this is Gregor Mendel. Gregor Mendel is considered the father of genetics. And he did his work in the 1860s, and he did his work in a country called Austria. Not Australia, Austria. Austria is located just south of Germany. And Gregor Mendel was a monk. He was a man of the cloth, and he studied an organism called Pisum sativum, or the common garden pea. And this isn't some rare plant. You can go to Lowe's or Home Depot today and buy pea plant seeds, and it's the same species. It's Pisum sativum. We eat this all the time. It's the garden pea. So Mendel studied 7 different characteristics of Pisum sativum, and he then studied how these traits were passed from generation to generation. Well, what characteristics did he study? He studied flower color and flower position. He studied seed color and seed shape. He then studied pod shape and pod color. And then finally, he studied stem length. So there's a handout that I posted in your folder that summarizes these characteristics. It looks like this. So this first column here are these 7 characteristics that we just listed. And then the second and third column organize the characteristics, whether they're dominant or recessive. What does that mean? Well, if I were a pea plant and I carry the gene for purple flowers, I would always express the purple flower color. Why? Because that gene is dominant. I might carry the white colored flower gene, but I don't express it. Since I carry it, but I don't express it, then that allele is called recessive. So this second column are all of the dominant characteristics: the purple flower color, the yellow seed color, the green pod color, and these are all dominant to these recessive characteristics here in the third column. Now the fourth column is really impressive. The fourth column is Mendel's data set. And so he crossed purple and white flowers almost 1,000 times. He spent 20 years studying this organism. This wasn't some weekend experiment. He spent 20 years doing it. Huge data set. If you then go farther down, you see that for the seed color and the seed shape, he has almost 8,000 data points. How's that possible? Because when he would cross a plant, the plant produces a pod of seeds. There's 4, 6, 8 seeds per pod. So his seed data set was always larger than when he was looking at the flower color or the flower position. So how did he cross plants? It's simple. Plants produce sexually, and the plants produce sperm, people call pollen, and Mendel would pass the pollen with a paintbrush from one plant to the other. So here are your two parents, right? So he's passing the pollen, and then to prevent other pollen from getting into this flower, he would cover the flower with a piece of cloth. And he would do this individually for every cross. So on the previous handout, where the data set showed that he made this cross 1,000 times, that means that just on that one trait, he did this transfer 1,000 times. Extremely patient. So this is Pisum sativum. It's the garden pea. If you like peas, great. If you don't, that's fine too. It's the pea plant. So when did he do his work? He did his work in the 1850s and the 1860s. He publishes in 1866. Now, people didn't realize that he was establishing the field of genetics when he published. They only thought that this was a very interesting study about plants and didn't realize that the rules that Mendel came up with actually applied to all living things. They just thought this is an interesting study about plants. Now, Mendel passes away in 1884 and has no idea the impact that he's made on science. In the early 1900s, there's a discussion as to whether the genetic material is DNA or protein. There's a British gentleman named Reginald Punnett, and Reginald Punnett rediscovers Mendel's work and presents it in this very simple little box that we now call the Punnett Square, and we'll talk about in the next couple of videos. And Mendel's work is then given international acclaim. He then gets credit for his work in the early 1900s, but unfortunately, he'd already passed away. So Mendel didn't get the credit that he so richly deserved. So when you think of Mendel's work, what he did, his basic experiment, was he took a purple parent and he crossed it with a white parent. These are two separate pea plants. And when he crossed purple and white, all of the offspring were purple. So what this told Mendel was that purple was dominant. So he then defined it as the capital letter. Now, he used R. People say, why did he use R for purple? Because when we define these characteristics, we try to use letters whose capital and lowercase look different. So you can't use P for purple because capital P and lowercase p look the same. So R has a different capital than lowercase. So since purple is dominant to white, he classified purple as big R. He then classified white as the lowercase r. And so what that means is that this purple parent is big R, big R. Well, why 2? Because we carry all of our chromosomes in homologous pairs. We get those chromosomes from our biological parent. So I get one chromosome from my biological mother and one chromosome from my biological father. I, as a pea plant, would then be big R, big R. Or if I was the other flower, I would be little r, little r. All organisms carry their characteristics in homologous pairs. There's always 2 copies. But then before we have offspring, you separate those 2 alleles and only give your offspring 1. Which is why in the F1 generation here, these individuals are all big R, little r. Why? Because this offspring got the big R from one parent and the little r from the other. Now, what Mendel did, he could have stopped there, but what he did was very simple. He then bred these organisms together. He said, what happens if I cross a purple, but he knew that these purple carried white? Well, how did he know that these purple carried white? because this purple offspring had a white flowered parent. So he knew that this organism here was carrying or hiding the white allele. When he then crossed these, what we call the F1 generation, he got a lot of purple offspring, in fact, in a ratio of 3:1, but the white reappeared. And people say, oh, the white skipped a generation. The white characteristic was there the whole time. It doesn't skip, it doesn't disappear, it just isn't expressed. So then the offspring here in the F2 generation, right, you get a ratio of 3 purple for every 1 white. And then this 705 to 224, these are the 900 data points that I talked about on the earlier slide. So the generations. Wherever you define the parents, that's always the P generation, P for parent, right? So then their children are called the F1 generation. Why F? For filial. Filial is Latin for child or offspring. So the first generation of children is the F1. Well, if the first generation of children is the F1, then it makes sense that the second generation of children is F2. So if you consider Dawn and I as the parental generation, that makes M F1. And then when M has children, her children will be F2. Now, where you define the P generation is totally up to you. You could consider my parents the P generation. Fine. Then that makes me F1. That makes M F2, and that makes M's children F3, and so on. So where you place the P generation is arbitrary. It's totally up to you. So when you go back to this handout, that's why that you see the P generation here. These are the parents. Then their offspring are the F1. And then the offspring of the F1 is then the F2, and so on. F1, F2, F3, you can go on for as long as you would like. So this is our introduction to genetics. There'll be another video following this talking about the Punnett Square and how we can actually track these characteristics from generation to generation. Talk to you soon.